An antigen is any substance that evokes an immune response in an organism. Pathogens are disease-causing agents. They, or their toxins, act as antigens and provoke an immune response from the host. The pathogen that we'll be following today is a bacteria. Bacteria are free-living, they do not require a host to survive, and are transmitted through food, water, air, or by direct contact. And they can be treated by antibiotics that target the cell wall or ribosomes, interrupting its life cycle. However, we will be following the natural treatment of invading bacteria through the human immune response. The first line of defense is any physical, chemical, or biological barriers to the entry of pathogens into the body. As you can see, there are many different components of our body that help to prevent pathogens entering. There's physical barriers, such as the skin, chemical barriers, such as the stomach acid that kills bacteria and pathogens that are swallowed, and biological barriers, such as the microbiome of beneficial bacteria growing inside the gut, as well as other barriers that you can see here. Pathogens can enter the body through many different ways, such as swallowing or inhaling, but today we will look at how getting injured by a cut enables pathogens to enter the body. The second line of defense. On every cell in the body, except red blood cells, there are major histocompatibility complex 1 markers. We'll call them MHC1 markers, and these enable self and non-self recognition. All of the cells in this scene have these markers, apart from the red blood cells. We can see that there are many neutrophils, and these are the most abundant type of phagocyte in the body. There are also mast cells present, and these cells contain lots of histamine, which will come in use very soon. Complement proteins are circulating the body as inactive precursors until they are activated. Currently, there are no pathogens present, and therefore no immune response is occurring. However, our person has accidentally cut themselves and transferred bacteria onto their tissue. The innate immune response, which is also called the non-specific response, now begins. Some cells may begin to automatically undergo apoptosis due to the physical damage from the cut. As soon as the bacteria enters the body, the immune cells begin to respond to minimize the harm the bacteria can have. The immune cells are able to recognize the pathogen as non-self due to its antigens on the surface of the bacteria cell. Mast cells begin to release the chemical histamine through the process of degranulation. This histamine causes blood vessels to dilate and become leaky. This increased permeability allows phagocytes, such as macrophages, to reach the site of infection. Up until these leukocytes reach the affected area, they would have been monocytes and then differentiated into the right phagocyte, such as a macrophage or dendritic cell, when they come across the bacteria. This increased blood flow to the area and swelling is the inflammatory response occurring. Complement proteins are activated and begin to assist in the immune response. They opsonize pathogens, making the bacteria easier to recognize as foreign to the phagocyte. They attract phagocytes through a chemoattractant. And finally, they form membrane attack complexes, also known as MACs, that assist in destroying the bacteria, causing necrosis. Neutrophils release cytokines, including interleukins and interferons, that amplify the immune response and recruit other cells to the infection area. The phagocytes at the site of infection, including neutrophils, dendritic cells, and macrophages, of which dendritic cells and macrophages are antigen presenting, then begin to engulf the bacteria through the process of phagocytosis. The process of engulfing bacteria continues. And once a neutrophil has consumed enough bacteria, it undergoes programmed cell death. These phagocytes do not last long and are not antigen-presenting cells. They simply die once they have consumed enough bacteria. The macrophages also consume the bodies of the dead neutrophils as well as the invading bacteria. Tissue repair occurs. Functioning cells or supporting connective cells begin to create new tissues to replace dead or damaged cells, such as the skin. The cycling of lymph throughout the body eventually collects the macrophages and they re-enter the bloodstream and lymphatic system. Even though these macrophages are leaving, others are still flooding to the area and the immune response continues. Macrophages travel through the blood and the lymphatic system via the lymph to arrive at the lymph nodes, which is secondary lymphoid tissue, and receive lymph from incoming vessels, and this is where lymphocyte activation occurs. As mentioned earlier, macrophages are antigen-presenting cells, meaning that they process and present antigens for recognition by T and B lymphocytes. This is a completely random destination for the macrophages, and they simply cycle through the circulatory and lymphatic system until they come across the specific T and B lymphocyte that they are to present to. Once at the lymph nodes, antigen presentation occurs, and the macrophage presents the MHC2 antigen complex on its surface for the T and B lymphocytes. The third line of defense now begins, which is also called adaptive immunity. We will first look at the humoral response, which is associated with the serum, the non-cellular part of blood, and involves the action of antibodies secreted by B cells, also called B lymphocytes. Overall, B lymphocytes' role is to recognize and bind to antigens. Each B cell recognizes one specific antigen, and then create/secrete antibodies to combat the invading pathogen. B lymphocytes can differentiate into memory B cells and plasma cells. Memory B cells will remember the antigen from our invading bacteria and remain dormant in the body until the antigen is presented again. When these cells encounter the same antigen again, even after years or decades later, they rapidly differentiate into antibody-producing cells, allowing a quicker immune response. Plasma cells. When stimulated by an antigen, some B cells differentiate into plasma cells through the process of clonal selection. Plasma cells secrete the same antibodies into the bloodstream. These antibodies go from membrane-bound antibodies to free antibodies when they are released. The antibodies then inactivate the circulating pathogens. These plasma cells only secrete a single specific type of antibody for dealing with a specific invading pathogen. The following is a structure of an antibody. Most of the molecule is made up of constant regions, which are the same for all antibodies of the same class. These are the heavy chain and the light chain. Variable regions form the antigen binding sites. Each antibody can bind two antigen molecules. The antigen binding site differs from one type of antibody to another. The huge number of antibody types is only possible due to the small variable region on an antibody. These antibodies can inactivate the invading bacteria in multiple ways. Antibodies can act as agglutinins and cause antigens to bind together, forming inactivated clumps. Antibodies also opsonize the invading bacteria, making them more obvious to phagocytes. We now look at the cell-mediated immune response. This is still part of the third line of defense. This response involves T lymphocytes. T lymphocytes will only respond to antigen fragments that have been processed and presented by infected cells or macrophages. When activated, T lymphocytes may differentiate into two different types: helper T cells and cytotoxic T cells. Helper T cells can activate cytotoxic T cells and other helper cells. This is done through the release of chemicals such as cytokines that signal to other immune cells in the body. It is these cells that activate cytotoxic T cells to divide and begin taking action against the invading bacteria. Cytotoxic T cells are highly effective killers. They will eliminate any infected body cells by releasing powerful cytotoxins when they come across a cell that carries an unrecognized antigen. They are also called T killer cells or natural killer cells. Ultimately, the cytotoxic T cell will eliminate infected cells and not the pathogen by itself, whereas B lymphocytes will eliminate the pathogen itself and not infected cells. This brings us to the end of the third line of defense. I hope you've enjoyed this video. Thanks for watching.